Okay, so without further ado, I will go ahead and introduce Dr. Michèle Studer. Do we have her ready and online? Okay. So Dr. Michèle Studer is the Interim Research Director at the Institute of Biology in Nice, France. And we are lucky to have her as one of our scientific officers. She is also a representative of NR2F1 France, our sister foundation in France, and a member of our international research team. Michèle is a researcher in the field of developmental biology, interested in the process of mammalian brain growth and differentiation. She has worked in the UK, US, Italy, and finally in Nice, France, where she has been running an interim team since 2009. While researching for genes involved in brain formation, she came across NR2F1 and never let it go. Today, her team is trying to decipher the contribution of mutations identified in patients to the pathophysiological mechanisms of this rare genetic disease. There'll be a quiz following this introduction, so I hope you're all paying attention. With the help of researchers, clinicians, geneticists, and above all, patients and their families, this is her words, not mine. Above all, patients and their families, the aim is to advance research into this disease and improve the care and well-being of our young patients. So, merci, Dr. Studer, pour votre dévouement à la recherche sur NR2F1. Thank you, Dr. Studer, for your commitment to researching NR2F1. Et c'est à vous maintenant. Merci.
Thank you very much, Carly. But I need the permission to share, so otherwise I cannot share my presentation. So thank you very much, Carly. And so I'm very happy and excited to be virtually part of the group. So just think that I am there with you. And I would, during my presentation, if you have any question, don't hesitate to interrupt me and ask me question. Okay, looks like I can share. Okay, so I got already a nice presentation. Thank you, Carly. So I will, this is a kind of overview of my presentation today. I will give you some kind of, go back to basic principles of NR2F1 and BBSOAS. I think it's important to always reiterate some basic principle. I will give you an overview of of the research strategies that we have in the lab. And then I will go through 3 different systems that we are using in lab in order to understand the impact of mutation in NR2F1, in how it affects protein modeling, and this how kind of impact that it has in living cells, and also what kind of mouse models that we are using as an in vivo experimental tool, and finally going to human IPS and show you the strengths and the limitation of organoids as an in vitro experimental tool. So just to remind you so you know better than me, the kind of heterogeneous symptoms that your children have that are not actually only, let me see now if I manage. Can you see my mouse?
Yeah, yes.
Yes?
Yes.
Okay. So that these symptoms, as you can see here, they are obviously the first is the developmental delay and the intellectual disability, different from mild to more severe. And then you have some problems, some morphological problem at the level of the brain. They have some visual impairments, but something that we also not talk much about, that you children different of the hypotonia have also, some of them have hearing impairment, some of them have repetitive behavior, and also oromotor dysfunction. So it's a very heterogeneous disease. It's not very different from other neurodevelopmental disease. As Sarah told you yesterday, this is a kind of frequency of the different symptoms. The vision impairment obviously takes an important role. as well as intellectual disability developmental delay. And yes, so it's a typical BBSOAS is part of the many neurodevelopmental diseases with similar features and heterogeneous symptomatology, as also Sarah told you yesterday. So quickly, NR2F1 is a gene. Here is located at this position, 5q14q15. You can see the chromosome here, and if we enlarge this part, this is how the gene looks like. The gene will, as we went through yesterday, this is the chromosome is kind of folded here in the DNA. So, and the gene as well. The gene will produce what we call messenger RNA, which is the lens, and go out of the nucleus here. It goes a messenger RNA, and then this messenger RNA will be translated in a protein here. And this is the length of the protein, and the protein needs to be properly folded in order to be functional. Now, if we have a look at the linear structure of the protein, you know this, we have mainly a DBD and a LBD for DNA binding protein domain and ligand binding domain. And this is important, the DNA binding domain is important to bind to the DNA, as the name says, so to kind of what we call target genes. And while the ligand binding domain is mainly, we think it's mainly involved in binding to what we call cofactors. And these cofactors are different from cell to cell, and this together with the targets will give the specificity of the function of NR2F1. So there are, as you've heard already, there are two major types of genetic mutation. So a simple point mutation, which will cause a change of just one letter in the DNA sequence, but this will have a consequence in the protein structure. And truncations, which are mainly deletion in the DNA that prevent the full NR2F1 protein from being made. So it's either a mutation at the beginning of the gene, so one of the two copies are not made, or you have nonsense mutation that results in a premature stop coding. So you have a shortened, you have a shortening of the protein, or a frame shift mutation, which is caused by a deletion or insertion in the DNA sequence and shifts the way the sequence is read. And this can also have a consequence, obviously, on the protein function. Now, as we have already said, the clinician tells us that according to the mutation, if it is a LBD, DBD, or we just lose one copy, or we call one allele, you remember we have two copies of one gene, then there is a kind of severity of symptoms, whereby when you just lose a copy, patient seems to be less severe than having a mutation in the LBD or the DBD. A very important concept is NR2F1 is called haploinsufficiency. So a BBSOAS is due to NR2F1 haploinsufficiency, which means as a product of a single copy, like here, because here green is the healthy single copy or the healthy allele, even if it's functional, it will be insufficient to allow normal cell function. So as I said, what a clinician found is that the increased severity of symptom depend on the position of the mutation or deletion, and this is what we called is a genotype-phenotype correlation, meaning is according to the genotype, there is a linked kind of phenotype. By phenotype, meaning what kind of symptoms the patient has. Now, if we go to the protein, this is how the protein looks like. So together with some biochemist, structural bioinformatician, They modeled the protein. So you can see here in pink, it's a DNA. In green, it's the DNA binding domain. And in blue is the ligand binding domain. Now, you can imagine that this is some of the, not all of the mutation of all the patients, are not all of them. You can imagine that if you have mutation either in the DNA binding domain or in the ligand binding domain, you will affect the function of the protein. And this is one thing that we are actually studying in the lab. Now, I told you that here you have the DNA, and that outside the DNA, you have a messenger RNA. Now, this messenger RNA, there are various types of tests whereby we can use this messenger RNA in order to understand where and how NR2F1 is expressed. And here you see that actually, so there are different levels of expression. So obviously, here the highest the number is here, it means it's expressed at higher level. So you can see that actually NR2F1 is not only expressed in the brain and in the eye, but also in other and many other tissues. And this is important for your children because it tells you that they might have problems also in all this tissue where the gene is expressed. It doesn't mean you will have, but be aware that the gene is quite expressed in many tissues apart from the brain. So, but if we go, but in the brain, where is NR2F1 expressed? So it's expressed very, very early. So this is a brain section, a human brain section at gestational week 8, so quite early. And here in black, you can see expression of NR2F1. You can see that it's expressed mainly in this region and this region. And this is another way, a little bit later, a few weeks later, with another technique. So this is, here they labeled the RNA. As I told you before, here it's the protein is labeled. And what is extraordinary is that it is expressed what we call in a gradient, which is low anteriorly, this is anterior, and this is posterior. So it's low here and high there. And this has probably a meaning because it is important when we look at brain structure. And what we expect is that the brain structure which are made probably most affected might be where the gene and the protein is highly expressed. What also is very interesting that when the brain convoluted, so you know that we have all this gyre and sulci, all this this convolution, NR2F1 is expressed actually quite highly where you have this convolution here, as you can see here in red, which again might implies that the gene might be important in the gyrification pattern, in the convolution pattern. Now, since the gene is expressed so early, and this is really a schematic about how our brain evolves, our brain develops, not only during embryogenesis, but also at birth, after birth, so from a simple tube to a very complex and fully developed brain, if any of the processes here that are important in getting this very complex structure is somehow affected, you can imagine that you might have malformation, and that this malformation will cause all the symptoms that BBSOAS children, but also other children with neurodevelopmental diseases have, such developmental delay, epilepsy, intellectual disability, etc. What are these processes? So here it is just a very simple schematic of what are the processes that are important during what we call corticogenesis, which is the development of our brain. And all this has been achieved thanks to imaging, to what we call functional genomics, to mouse models, etc. So the main processes during corticogenesis are what we call neurogenesis. First of all, are kind of proliferation. The cells have to divide and proliferate, okay, to become from 1 cells, 2, 4, 8, etc. So this is the first step, proliferation, here indicated with this arrow, before becoming a general neuron. Then this general neuron has to migrate. Migrate means it has to go into a place where it has a function. Then he has to acquire an identity, not a just simple neuron, but a mature neuron. And this mature neuron might communicate with other neurons in order to build a kind of functional circuit. So what are our research objectives in the lab? So first of all, we aim to understand the genotype-phenotype correlation. So how the different mutations impact on a phenotype, so on the symptoms of your children. Then we would like to reveal the impact of the different mutation on the gene and the protein function, study the developmental consequence of the different mutation, and also find an experimental model that is adapted to test future therapies. So first of all, you patients, your children are the most important part of all the research. And yesterday has been said multiple times, we need your clinical data, which have been registered in a single register. And obviously, the associations, the foundation are very helpful for that. Then what we do is modeling in silico the protein. So I show you how how the protein, the normal protein looks like. So this is a model of the protein, a structural model. And now we want to see how it changed in the presence of the mutation. And we want to validate this in cells, in normal cells. And then we want to insert some of the mutation in animal model in order to mimic the NR2F1 haploinsufficiency. And then we would like to go into a more complex model, which is a human model, and which is obviously more similar to us and to the children, in order to better understand the impact of the mutation on human cell behavior. So this again is what we are doing at present: protein structure, cellular in vitro assay, in vivo mouse phenotyping, and in vitro human iPSC for pluripotent stem cell phenotyping. So we start with the first two. So I showed you again, here is just again the modeling of the protein, the DNA here. See how it binds to the DNA. And here it is the ligand binding domain, the DNA binding domain, the ligand binding domain, and a potential cofactor. So here you can see they put the different mutation, some of the mutation in the DNA binding domain. And here what we are studying is how it impacts on binding to the DNA. Or if we isolate in silico the ligand binding domain, the idea is to see how it impacts the binding with the other cofactor. One is itself, one is its homolog NR2F2, and there are also other nuclear receptor or other factor that have the ability to bind to NR2F1. So to do this, you have to isolate the different domains and kind of modeling them in silico. Now the question is, do this mutation impact NR2F1 protein stability and function? So in terms of protein stability, our collaborator, biochemist, they actually did a full list of how the mutation affects protein stability. This is just an example. And you can see, for example, from here, from red to green, that these mutations are affecting very highly the stability, less and almost none. So there is not all the mutations have an effect on protein stability. So this is important to know, some more than others. Then what do? We take some of the mutation and make them express in a cellular system in which NR2F1 is not expressed. And then what we look at? We look if the cells are viable, so if they survive, is in good shape, how they behave physiologically, how the protein is localized within the cells, and then we try to correlate this with the patient symptom together with the stability. So I give you a kind of an example. So this is schematized, this is the nucleus, this is the DNA here, and this is how normally NF2F1 is bound to, a normal protein is bound to the DNA. So we found that, for example, a mutation here, which is a truncation, in this case, the mutated protein cannot enter the nucleus, so it stays outside, which means it cannot do its proper job. In other cases, some of the mutation give a protein whereby they are partially localized in the nucleus, but partially outside the nucleus. But if they enter the nucleus, they will bind to the normal protein, and this can actually have an abnormal function because this protein is mutated and then will not behave like its healthy counterpart. So, for example, just here, a protein mislocalization outside of the nucleus, inside of the nucleus, can have an abnormal cell function, and this is what we saw. We saw the cells were not happy. We saw that the cells were not proliferating properly and were dying. Now, this is, you don't have to read it, but this is just a kind of table that we draw at the end. And to kind of show, this is to show you that actually some of the worst in terms of effect, in terms of protein and in silico, in cells, actually correlated with the majority of the symptoms that the patient having this mutation have. And so we can kind of draw a severity score whereby, for example, this mutation here are the worst compared to this mutation, which seems to, in our system, to affect cells and protein stability less. Now, this is fine, this is protein, this is in cell culture, but what's happening in vivo? So obviously in vivo, we cannot use human being, so we use the mouse model. So as you know, the mouse model is a very fantastic animal model for modifying genes, and the way now genes are modified in the mouse genome is with this CRISPR-Cas9 technology. You might have heard this on TV or in journals. They're called also molecular scissors, and they cut and repair the DNA, and are used for gene therapy, in agriculture as well, for mouse model cells, and so on. And they're allowed to introduce mutation. In this case, we did it in mice. So what kind of mouse model we have? So this is, for the moment, the mouse models we are studying. Actually, not all of them. This is the first one that has been studied in the DNA binding domain. It has been published in 2020. We asked if we could get this mouse in order to compare them with the others, but unfortunately we had no response. This is really a pity, I think. Nevertheless, we now have, together with Christian Schaff, we have 3 mouse models, one the mutation, the DBD, and 2 with a mutation in the LBD or between the DBD and LBD. And this, the mutation in patients, they show kind of different severity of symptoms. So we thought this was important to compare them. So we go back to the question. All these mouse models need to be compared to a healthy control, so to a normal mouse which has 2 copies of alleles which are normal. We use also a mouse with a deletion, just a deletion of 1 allele, and this mice where we have 1 allele with a mutation, and actually this mutation produced a protein, but the protein is not completely normal. In this case, so here you have 2 alleles with 1 normal protein, 1 abnormal protein. Here you have only one normal protein working. And for the moment, I'm going to show you some results of these models while we are together with Christian, we are working on these other 3 models here. Now, so when we talk about haploinsufficiency and our mouse models, when the mouse is kind of healthy, you have high expression here in red of the protein. So this is here where the protein is expressed in the brain. When you have a heterozygous, only 1 allele is working, you have a diminished production of protein, so here red is less strong. And when you have no allele that is working, then you have no protein at all. So the condition of the patients are here at the heterozygous, but it's also important to look at the homozygous because it helps to see what, even if we don't have patients, it helps to see the kind of extreme phenotype where you have no protein produced at all. And this helps us also to better understand what's happening here. So we use the animal model, mouse model to study NR2F1 haploinsufficiency. And what we study mainly is the brain and the eye. And we looked for cerebral malformation, circuit dysfunction, behavioral disorders, if they have epilepsy or not, for example. And also in the eye, we looked for optic disc defects, retinal anomalies, optic nerve atrophy, ocular visual deficiency. So I give you some example of what we found. As here at the bottom, you will see some of the publications. So if you're interested, you can go into them. So if we took the eye, so this is the eye, what we call the retina. This is the lens. Okay, image, it's from here. And here the information goes to the brain. Let's say it is here. So this is the retina, and in the retina, the first cells that differentiate are called ganglion retinal cells here in red, and they are the one that are producing, forming the optic nerve. Along the optic nerve, you have other cells that are called astrocyte or oligodendrocyte, doesn't matter. Now, in humans and in mouse, NR2F1 is expressed here, so you can see here in red, quite strongly only mainly in one part of the retina, but also here along the optic nerve. So what's happening if we decrease the dosage of NR2F1 in the eye? First of all, what we found, this is a kind of picture of a 3D picture of a mouse brain. The eye is here, and this is reproduce the eye at this level. And you can see here, this is the optic nerve, and here. First of all, the optic nerve, you can see it here from here, also section is thinner. So not surprising, we have a hypoplasia of the optic nerve. But second also, which is very interesting, is actually a little bit distorted, as you can see here. And what happens is that the axons here, so the bundles of the optic nerve, instead of going nice middle of the nerve, actually they tend to be stuck more at this part of the nerve, as the ventral part. So they are elongated, but here they are kind in a wrong position. Then what happens in then? Then the axon here, so this is a neuron, you have the soma, you have the axons. The axon, in order to be functional and to transmit a stimulus, like a visual stimulus, it needs to be embedded by what it's called myelin, as you can see here. The more myelin you have, and you can see here, the better it is, because the axon will be insulated and the stimulus will go very quick from one neuron to the other. So what we found is actually in the mutant mice that the optic nerve, so this is a section of the optic nerve, here in black are the sheets of myelin. These are diminished. There are less myelin. But we saw this also actually in the brain, not all in the optic nerve. So that the myelin is not well constructed, as you can see here, and the sheets are not very compact. So there is a problem of myelination. So first, a problem of guidance of the axon, of pathway of the axon. Second, of myelination. What else? So if we look at the retina, so you can see here is just a diagram of the retina as the different cells. So the one I talked before, the ganglion cells, they have their axon there, going through the optic nerve and then going to reach the brain. But there are other cells that are important for vision. Now, your children might have gone through an optic coherence tomography or OCT. And this is a very interesting technique. It's non-invasive. It's a diagnostic technique. And that gives you in vivo cross-sectional view of the retina, as you can see, of the different layers of the retina and its composition. Now, in a study that we did with some British ophthalmologists, we found that the majority of the patients have problems in some of the layering. So we looked in the mouse as well, and this is also what happens in the mouse. For example, here, the ganglion cells are less. Even if it's not super severe, they are less. These cells here, the amacrine cells, are more. And here, these cells, the bipolar cells, that are still important for transmitting the stimulus, the visual stimulus, are here less. So the phenotype is not super severe. Also in the OCT, it's not super severe, but it's still there. So what's happening in terms of function? Because we talked yesterday also with Lee that there might be a vision function, and most of your children, they carry actually, as you know, glasses. So in the mouse, what did we find? If in the mouse we give a light, a flashlight, and register at the different, parts of the visual pathway, if the stimulus is going through, the answer is yes. The mouse, it sees, but the latency is higher, which means that the stimulus is going through properly to the brain, but slower, okay? And if we challenge the mouse in seeing kind of stripes, more and more thinner and thinner, what is called testing visual acuity, and some of your children might have done through a visual acuity test, again, we find that the visual acuity is decreased. So in the mouse, what we found at the visual stimulus goes through, but it's slower, and they are not super good in discerning different patterns and having a high visual acuity. So this is just to tell you that it's very important to go back and forth with patients, having the mouse and look at the patient, having the patient and look at the mouse. This helps a lot in better defining what's happening. In this case, this is an example of what's happening with the vision, but and to better pinpoint the major defects that are in the mouse and in the patient, and to go even more in the depths and find really the kind of molecular cause at single cell cause. Now, the eye again, and as we told yesterday, the eye, the optic nerve will go to the brain, and we go to the visual cortex, which is the cortex here, which is the most occipital, the most posterior, anterior here, posterior. I remind you that here is where actually NR2F1 is highly expressed. So we talked about cerebral visual impairments yesterday. So just to remind you, it's a form of visual impairment caused by the brain, which is not able to process information for the eyes. It doesn't mean that it is not necessarily caused by eye malformation, and usually it affects the posterior visual and associative pathway to the occipital, parietal, and temporal lobes. And yesterday, they showed you yesterday, there are kind of streams. Here, a dorsal, this is the visual cortex, and there are information that are going in this way, and this is important to process where is the object, and then what it's called a ventral stream to the temporal lobe of the brain, and this is important to understand what is the object, where is the object, and where is the object. Obviously, this is very simplified. Now, together yesterday, we showed you, so together with the American groups and our French and Italian group, we did some magnetic MRI imaging on several BBSO patients, so American patients, European patients, and we kind of agreed about the majority of the features. Here, for example, we see very clearly that there is a reduction of the optic nerve and also of the chiasma. The chiasma is where the optic nerve cross, because you remember that the left eye goes to the right hemisphere of the brain, and the right eye's information goes to the left hemisphere of the brain. What we saw also is that there were problems here in how the brain was convoluted. And to our surprise, we found that actually there is a higher gyrification pattern, that there are more kind of convoluted gyra that are either enlarged or reduced but more frequent in regions actually which are the more occipital region where the visual stimulus is arriving, that are important for processing the visual information, but also actually there are also some regions important for language. And as you probably also know, this is very consistent in your children, and it happens quite often that they have an abnormal corpus callosum. So you can hear the corpus callosum is important to actually connect the two hemispheres of the brain. And with an abnormal corpus callosum, there are very many, it's very known, there is a lot of literature, there are different behavioral disorders, like problem in coordinating motor activity, a deficit in cognitive processing time, abstract reasoning, short-term memory, and social linguistic deficit. So having a thinner or abnormal corpus callosum can also have an impact of the symptoms of your children. So what's happening in the mouse? Well, in the mouse, we don't have a super convoluted brain, because the mouse brain is not convoluted, has no gyra, but we have an enlargement here of the structure. And again, an enlargement of the occipital structure here. Here in red, again, is expression of NR2F1, highly here at this level. And we saw that the main reason of this is that it's because these cells here, which are called the progenitor cells that are important to enlarge the pool of future neurons, future neural cells, tend to proliferate too much. Too many cells, and this is because is due to the cell cycle, which is shortened. The cell cycle is important to say to the cells when it has to divide. And if the cell cycle is shortened, so they will divide more. So this we found as cellular and molecular mechanism to explain this enlargement of the occipital part of the brain in the mouse. Now, is this happening as well in human cells? So obviously we cannot work on fetus, as you know, so this is why we decided to move on induced pluripotent stem cells and to do 3D organoids. So quickly, so pluripotent stem cells, they derive from somatic cells, which can be fibroblasts from the skin, so a kind of biopsy from the skin, but also simple blood cells. These cells, somatic cells, they will be reprogrammed, meaning that their fate will become kind of embryonic, back embryonically, will become kind of naive thanks to a cocktail of factors. And this is why they're called induced pluripotent stem cells, because they have been induced to become stem cells. And pluripotent. Pluripotent means they can become different types of cell types. So not only neurons, but they can become also heart cells, they can become liver cells, whatever you want. So then you need a cocktail of factors to differentiate, in this case, of neurons. Neurons can be 2D, flat, or 3D, we'll see in 3D. And these are a wonderful platform to test different drugs and to test different drugs on patient cells. So the idea is to have in the future a personalized regenerative medicine, whereby we can, an industry mainly, will use patient cells in order to test drugs. Now, how do they look like? So iPS cells, if we differentiate them in 3D, they can be flat or they can be 3D. Here, there are little kind of aggregates of cells, as you can see them here just by eye or just a little bit by the microscope. And actually, what is superb of this system is that they form a 3D structure, meaning like it's in the normal embryonic brain with different layers. So here you have the progenitor cells. You remember I said that in the absence of NR2F1, these cells proliferate a lot before becoming neurons. And here you have the neurons. So this is a good system to study what happens. And now we set up the system and we can, you see this is called a rosette. Here is a rosette. This is how they call them, rosettes. And you can use different markers to understand what are the progenitors that eventually become wrong, what are the neurons that eventually becoming wrong. So we are in the process now in studying the effect of haploinsufficiency of NR2F1 in a heterozygous and in a completely absence of NR2F1 in these cells. And these cells were kind of produced by CRISPR-Cas9, the kind of molecular scissor I talked about. So this means that we have some control organoids, and then these control organoids, NR2F1 has been altered, and then we can understand what's happening in the mutant organoid, compare them to the control. The second strategy, which is obviously the most interesting one, is to take have blood cells or whatever fibroblasts from your children, to reprogram them, get iPS cells, and then go to mutant organoids. organoids. So this, which again will be directly from the patient. Now, the problem of this strategy, which is obviously very powerful, is obviously, and this will give us the opportunity again to understand how the different mutation affects the behavior of this IPS and this small kind of little cerebral organoid. The problem of this strategy is that we cannot use as control any type of IPS because the genomic landscape of your children may be unique. So we need to kind of correct the mutation that is in the IPS, and this will be controls. So this is at the moment where we are a little bit stuck at the level of this patient IPS, because as I said, IPS we have IPS and we have quite a lot, and there is in the biorepository more and more. This IPS which harbor mutation, they can be differentiated and we can do all this wonderful thing. However, we need a control. And here we are kind of stuck because we need an isogenic control. And reverting the mutation is simple, but not as simple. And it needs some funds, some helps. And I hope eventually that with a grant, we can achieve this. So I'm finished now. So just to sum up, so what we are now using in order to understand the phenotypic heterogeneity of BBSOAS children, as I show you, it's by using in vitro approach or in silico, in vivo, and human organoids. And this all thanks to the clinical network, to the different association and foundation, the neuro-ophthalmologist, and the reprogram facility, and obviously with combined this non-profit user consortium that hopefully will also, it's highly contributed to BBSOAS. Last but not least, yesterday, somebody asked about epilepsy. So I'm not really working on epilepsy, but I wanted to make you aware that this year came out a paper whereby some Chinese researcher found that perampanel, and here I just summarized a little bit, which is an anti-epileptic drug, seems to work quite well if together with other drugs. So what they took is 20 patients. They had a cohort 20 patients, 60 of BBSOAS, right? 60% had this kind of like epileptic features, as you can see here. They used several anti-seizure medication, as you can see here, but seizure were controlled only in 50% of cases. What they found in 6 new cases, that the children became completely seizure-free after perampanel, which is another anti-epileptic drug, as an odd treatment. And this is the dosage. So what I would recommend is eventually look at this paper and talk to your neurologist or to your clinical neurologist, and eventually think for the children that have epilepsy, eventually to think about using this drug as well. And here I'm finished. This is the group, the team in Nice, the different people, our collaborators from clinicians to basic research and biochemists, our funding at the present. And again, thank you very much for the association and NR2F1 France Association and NR2F1 American Foundation. And thank you to your attention. Are you there?
Yes. Oh, is this working? Yes?
Let me stop. Okay?
Thank you so much, Dr. Studer. That was quite informative. I think we'll be having another quiz. I hope you all got all of that. And that's why we are so grateful that somebody like Dr. Studer is working on NR2F1 and for our kids.
So if you have any questions,
so if we have any questions, do we have any questions for Dr. Studer? I have one. Oh, sorry. Okay. I have a quick question before I forget. That paper and that drug that you mentioned, is that paper free access or?
Yes, yes. I have
Okay.
sent it to Melissa, actually.
Okay, so we will share that out with the community as well, just for your, yeah.
Our son had an MRI as a result of his seizures, and we've been told by our neurologist that his MRI is normal. And I'm wondering, is what you were talking about with the myelin, is that, or in the enlarged gyra, is that something that would be normally visible on an MRI? And so is it?
So the gyra, the gyrification, yes, but you really need somebody specialist, because this is also what Viral said yesterday. A lot of the MRI that were obtained from normal, testing of MRIs that you get routinely. A lot of them, I send them to a neuroradiologist who is used to really look carefully at MRI, and it's a subtle, this is what they found. So you need a specialist. Usually when you do an MRI, they see the most obvious thing, which is what I showed you, the optic nerve, hypoplasia of the optic nerve, and the corpus callosum. The gyrification is something that you need to have a really kind of specialist eye. For the myelin, this you don't see it by MRI. Well, you can see it by MRI as well, actually. Again, you can see it, and this is something I think we need to do more carefully. Again, you need a really specialist of that. The one that I show you, first of all, were not MRI. They were electron microscopy images and on mice. Okay, but if I remember well, in some cases there were mentioned that there was a hypomyelination in the brain. So this is something I think we need to look more into details. Yes.
Okay, do we have any other questions in the room? We do. Elodie from France. No, well, originally, but from Montreal.
Hello, Dr. Trudel. I don't know if you have an answer, but when do we expect some treatment to begin?
Big question.
Well, I mean, what is your best scenario?
I cannot say.
Okay, so you have no clue, or?
No.
Okay.
You have to understand something which is very important. It is a neurodevelopmental disease. Okay, I've tried to show you that things happening very early. Okay, so all what you see in your children at birth and after birth is actually due to defects that happened in utero, so while you're during your pregnancy, but you're not aware of. So it's very, very difficult to cure the major cause, the primary causes. If the primary causes, as I show you, it's that the progeny proliferate too much, you're not going to cure that. What we can do, what is important, is to diagnose the disease as soon as possible, and is to try to treat the symptoms of the disease as soon as possible, which means the first years of life. You see, this why? Is this because the brain is very plastic? So this is where you can act. So the idea is to try, what is the rationale? The rationale is to try to find the embryonic causes in order to see what will be the effect and then treat the effect. So for example, one of the consequences of what happens before is epilepsy, for example, right? So you want to treat epilepsy, but you want to know why you have epilepsy, at what level, okay? So the only thing we can do, so research is important to understand the causes of the defects, and then we can go later on and try to diminish the effect of what has produced the major defects that happened embryologically after birth. I hope I answer your question, but I cannot say it's going to happen next year, in 10 years, in 20 years, in 5 years, and so on. Don't forget that BBSOAS is a rare disease. Okay, our force is to work with other neurodevelopmental disease, because all of them makes a 3% of children disease, okay, which is much more than talking about a 0.001% of BBSOAS. And because these diseases are neurodevelopmental diseases are very similar, the force is to work together as a community on different neurodevelopmental disease and help us to find therapies that can be used after birth, okay, and exchange information, hardware therapy that eventually can be used in different diseases. So this is a strength, okay? I hope.
Thank you. Thank you very much, Dr. Studer. That was probably the best answer anybody could give. It is $1 million question. That's why we're all here.
Yes.
So we do have some questions online, but for the sake of time, I do apologize. We're going to have to, we will email them to Dr. Studer and she'll get back to you. So Claudia, Victoria, John, Danae, and Victoria. Again, yes, we will get you those answers.
Yeah, just tell them to send me by email the question and I will answer the question by email.
Perfect. Okay. Thank you.
And sorry for the delay of this kind of technical problem. Usually it works very well.
Of course. No worries. Thank you.